So I think we can get started. Welcome to ISR Colloquia. For those of you that this is your first time, we have this fantastic seminar series at the beginning of each month, the first Wednesday of the month. And the idea here is to have interactive discussions and questions and answering and learn about the diverse nature of ISR through this seminar series. So thank you for coming. Today's speaker is my very good colleague, Professor Timur Horuyuchi. Timur received his bachelor's degree in electrical engineering in 1989 and PhD in computational and neural system in 97 from Caltech. Following that, he did a postdoctoral associate at Johns Hopkins University. Timur joined University of Maryland in, I believe, fall of 99. Yeah. And he has been part of the microelectronics group in the electrical and computer engineering department, as well as he's been a faculty member in ISR all these years. Timur is a co-director of the Computational Semiconductor Systems Laboratory, and he's also a member of Neurosciences and Cognitive Sciences program at University of Maryland. He is also one of the key players at the Telluride Neuromorphic Engineering Workshop that takes place every summer in Colorado. It's a very, very known and stimulating workshop that he's one of the key members. And he's also a member of the IEEE. Timur's general research interests are in computational neuroscience and implementation of neural circuits and architectures in VLSI-based processors. And today, he's going to tell us about some of the latest and exciting work that he's doing in his group. So, with no further ado, Timur, please.
Okay, thank you. So, what I wanted to start with is just by pointing out that I've actually changed the title. There used to be a 3D here. It was a bit more aspirational than something we were able to achieve or something worth talking about to you now. Many of you are actually already familiar with a lot of the material I'm talking about. As Reza mentioned, a big part of my lab's research is about trying to understand how biological systems go from neurons to behavior, which is also known as ecology. We consider ourselves silicon neuroecologists, trying to go from silicon neurons all the way up to behavior, trying to understand this larger perspective. We use robotics really not as the end goal here, but really more to sanity check, reality check our ideas of the neural systems in closed-loop behavior. In particular, the VLSI is good for real-time behavior, and we believe that it's an important perspective to have when considering neural systems and trying to understand the behavior. So we try to go more towards real-time behavior, even if it is simpler in particular. And so as you can see from the slide, really, we're interested in bat behavior, bat neurobiology, even building physical structures that are echolocation systems, and looking at platforms in which we can test these ideas. In particular, I'm working collaborating with Sean Humbert on the quantum rotor system, and I'm also interested in some ornithopter-based systems. But essentially, the lab designs VLSI and tries to understand computational neuroscience literature. So before I really jump in here, I wanted to point out some of the people that are part of my lab currently and have contributed in the last year or so to the material that we're talking about. Certainly there are many, many other people that have contributed recently. And so I wanted to point out, first and foremost, Tarak Masood, who just graduated because PhD is now working for Intel up in Portland, Oregon. And I've been a graduate student Matt Runchi, 3 graduate students in recent past. Peter is currently working on it. Peter and Tim did a merit project this summer on attentional systems in sonar. And Peter is doing a 499 project with me now, as is Roseanne, who's also doing a 499 project with me. My primary collaborators are these are all ISR faculty, in fact. P.S. Krishna Prasad, who does work in controls and has brought a lot of mathematical rigor to my own thinking. And of course, Cindy Moss, who runs the BAT lab, and in which we investigate both behavior and neurophysiology. And with Katrina McLeod over in biology, we're going to do a number of simulation work on synaptic functions based on her experiments. So the lab in the past has done a lot of work in terms of sensory processing with respect to sonar. We've worked with narrowband systems, as primarily what we'll be talking about today, although it's not really a sonar talk per se. We've worked with broadband systems, and developed broadband ultrasonic cochlea, and thought about signal representations. We've done extraction range and azimuth out of the sonar systems using the physical structure of the head to produce localization cues. We work on instrumentation for putting microphones to record vocalizations in flight. And at a higher level, we've been thinking more biologically plausibly in terms of proposing architectures for things like avoiding targets that you see, or looking at targets that you see, which is more about looking for specific things, so it's kind of a top-down perspective on sensing. And what I'll be talking more about, which is really about spatial memory, spatial cognition, or spatial behavior. And this is a particular passion of mine right now, in the sense that it's this larger question of how do you go from just sensing what's in front of you to a larger perspective in terms of understanding the space around you, in particular for navigation. So there's kind of an outline of what we'll be talking about here. So first of all, I'm basically review a number of literature studies that focus on this basic idea and inform some of you that may not already know a little bit about what is known about space in animals and the unique perspective that this system really has. And then we'll talk about some efforts in neuromorphic VLSI to try and build and model some of these ideas, and discuss a little bit about what we've learned. There aren't always so many messages or lessons that we can bring back directly, and I'll try to talk a little bit about that. And then beyond, how do you actually use this kind of a system? This is one of the questions that always comes up in terms of, well, we'll talk about place cells in particular. So I'll mention some of that at the very end. So I said that I removed the 3D from the title, mainly because 3D is the primary interest that I have now, and we haven't made as much progress towards that as we would like to at the time I proposed the title. So here's our hero. This is the big brown bat. That's, for those of you who know, it's a ferocious pan. This is a big brown bat. Not so big, but it is bigger than the little brown bat. So I've noticed that certainly over the years, this bat has gotten cuter and cuter to me, whereas when I first joined the lab and I encountered bats, basically the bats didn't always look so cute as they do now. So this is a video just to give you some idea of the behavior, just get a concept here. So up here is a bat that just loves this little corner, the old configuration of the laboratory. And you're looking at through an infrared camera that's digitizing the flight of the bat. There's a microphone that we designed and placed on the head, and so you'll be hearing the vocalizations. The video has been slowed down by a factor of 10, and the sound has been slowed down by a factor of 10, and they're synchronized. So the idea is that because bats use ultrasonic vocalizations, you can't directly hear them. By slowing them down by a factor of 10, you can now hear them in the audio range. I'm not actually sure to test this sound. Play it twice. So there's actually a worm hanging from a thread here that and so what you should be thinking right now is that this is a coordination, sensory motor coordination, a very complex one of navigation, about target tracking, about attention, about vocalization, changing the vocal structure, all in the service of capturing this worm. Albeit the worm was stationary. But in general, this is a very large problem and has a lot of applications to a lot of engineering concepts, and more than even that I had thought about before. So the question I'd like to basically pose today, or to have you start thinking about, is this really lightweight sort of question. How do animals keep from getting lost in their environment? Very general statement. This is true for robotics too. If any of you have actually, so on the surface, this question seems really trivial. It's not, we know where we are. We look out in the room, we see where we are, we turn around, and it's still there, we know it's still there. And we have this idea that we understand the world enough that we can probably close our eyes and do this in the dark. But if you think about programming a robot to actually do this with sensory data, you have to construct some sense of what is where. And this larger question of where are you now? And so this is kind of wrapped up in this idea, this topic called SLAM, or simultaneous localization and mapping. And engineers have approached this problem very, very, very differently than what we've found in biology. And while there have been a lot of efforts to try to understand how biology does this, the experiments that are really revealing some of these things are still very, very much in flux. So let me pose a more concrete sort of statement, which is to say, if an animal didn't have GPS, a compass, somebody gave it a map, what would you need? What are the capabilities that would be behind something like this? And the first thing that always comes up is, well, okay, if I have a way to do dead reckoning, I'm going to remember exactly, I'm going to maintain a position, an estimate of my position in space relative to where I started. I'm going to track the translation. If I turn, I'm going to try and estimate that turn. I'm going to calculate. So I'm always carrying around these two final numbers about where I am with respect to the origin. Now, when you do this kind of thing, and people do this all the time in robotics, usually the questions of accuracy come up. The very first thing, gyroscope and everything like this, estimating velocity. How do you do all that? Well, that is a way, and this is what I'll be talking about primarily today, although we won't get anywhere close to the questions of accuracy in particular. You could approach this by thinking about action sequence. You could say, well, I'm going to remember what I did, and then I'm going to turn around and go back. Because the key thing here is, when you fly to a new city, a taxi drops you off at a hotel, you open the door at night, you say, I need to get some dinner. You check Yelp, or somebody says, go over there. You travel out into the streets, and you get to your restaurant. If you can't find your way home the very first day you try this, right? If you're a bat, you're probably going to land somewhere and eventually starve, probably. You need to find your way home the very first time, every time, right? The minute you don't, potentially you're going to be in a lot of trouble. So the issues about action sequence is the invariability. You may not be able to say, okay, I turned 20 degrees here, and then I travel this far, and be able to know how to turn that around. That's actually fairly difficult. It may not be invertible at all, meaning get on the bus, sit in the seat, and then wait 16 minutes, and then get off the bus, right? You may not be able to turn around and go back, because the bus doesn't go backwards that way. Stationary, the bus may run at a different time. The bus may not be there. Landmark detection. So this idea that maybe I could go by waypoints, right? I can remember the action sequence of landmarks. The big problem here is this big question of, what is a landmark? Right? I'm going to turn left at the pigeon and turn right where the water, the flower is growing. If you don't realize that, you need to have a lot of experience in the world to understand that a landmark is a landmark. Because a landmark needs to be something that's visible from far away or detectable at very far distances. You need to know that it's unique or unique enough. Or you need to understand how unique it is, right? It is helpful to know that if you detect a car, you may be in a parking lot. The car is not unique, so it's not really a good landmark, but it's a clue. And the last of all is this idea of the internal map. So this idea that more like an engineering approach, where you say, okay, I'm going to allocate some space in my memory, and I'm going to start labeling or sticking things I encounter in this map. And I'm going to either have to have it be metrical or something. But the big problem there is this idea of scalability, which is that presumably you're using the same navigational system for me to get out of this room, or within this room, or even within this space, as I am if I need to drive home. Right? The scales are hugely different. And presumably using very, very similar types of navigational or spatial cognition to do that. And the minute you start allocating resolution and allocating little boxes for specific spaces, even if it's multi-scale, you're going to have all kinds of problems with storage. And so people have thought a lot about this, that maybe the world and what you store is going to be a little more topographical, not going to be metrical. That I just need to know that I go that direction until I see something. And so I'm going to get a topology of the world. I'm not going to have a metrical map. And that allows me to store a lot less. So in the brain of mammals, a number of cells have been discovered. And what's interesting about this is that, for the most part, in neuroscience, from the very beginning, almost all of what people have understood about sensing capabilities have come in the form that the neurons that fire are usually in the reference frame of the sensor. If you've studied visual processing, in general, this is, we call this retinotopic cortex. And you find everything visual seems, for the most part, to be in more or less retinotopic coordinates. You find that you don't find that much translated from one coordinate frame to another coordinate frame. And so what was really exciting in 1971 is when John O'Keefe discovered these things called place cells. And this is the first instance that I'm aware of where a cell is now firing with respect to the external world in its reference frame, outside of the animal. The animal somehow separated the response of this particular neuron from its own body coordinates, from its own sensing coordinates. Okay? There are a lot of ways you can argue around this that maybe it's not quite true, but for the most part, here's a neuron that, it will turn out, and we'll discuss it, that fires when the animal is in a particular place, loosely defined, a location in the room, in its coordinates, what we call allocentric coordinates, in the world frame, and not with respect to the body. It's very easy to think about a receptor that's going to detect something 20 degrees off from where my eyes are pointing. And then secondarily, in 1984, was the discovery of head direction cells. These are cells that fire when the animal is pointing in a certain direction in a compass-like fashion with respect to the environment. So now I have kind of almost the building blocks for a map, right? A map and compass almost. Location and a compass. But it's not a magnetic compass for the most part, as far as we know for head direction cells, although there's evidence for magnetic compass capabilities in some animals. And then, after all of this, a lot of modeling that followed, grid cells were discovered in 2005. And it trashed a lot of the models that people had been developing prior to that. Not that they're all wrong, it's just really more that it really threw a monkey wrench into the works. And so since about 2005, there's been this scrambling of models. And the most exciting thing right now, I suppose, is that there's two camps of models and we still really don't know which class of models is going to be correct. But we will eventually find out. So the other exciting thing is that these types of cells have been found in an ever-growing list of different animals. And of course, I'm here to think about bats in particular. Most of these studies have been in rats. And rats are very good at navigating two-dimensional environments. So there'll be an interesting question. So this is a fun graphic, which is common. The place cell, like I suggested before, this is representative of the experiments that have been done, and I'll be showing you more of. A place cell is a cell that fires in a particular location in the environment that's relatively localized. It's usually, well, it's often multi-bumped, one location, although that's not strict. There are head direction cells that no matter where it is in the environment, when the animal is pointed in a certain direction, with respect to the environment, it will fire. And so it's like a little compass. And the most exciting one and the most baffling and intriguing one is this grid cell, where if you overlay a triangular grid on the environment, then the neuron fires on its vertices. And it fires in a repeating periodic pattern. It's one of those things where you think, that's crazy. What's going on? And so we'll get into the models that discuss some of that. And how could it possibly be used? So the head direction cell is pretty much what the title says. You have neurons in the postspiculum, which is an area that's very closely physically related and connectively related to the hippocampus, which is the area we'll be talking about. These neurons have tuning curves where they're active, meaning they fire spikes, they're firing digital pulses, effectively, that's tuned for a particular orientation in space. So here's an example where they've collected 4 that are nicely spaced. In general, there's a nice broad distribution of different neurons. What's interesting is that this cell, however it gets its cues for its orientation in space, will continue to fire even if you flip off the lights. Okay. It still knows its insert direction. You get it set up. And then you turn off the lights, it's still going. It still knows. It'll make a turn, it'll come around, and it'll still have an estimate, but it may start to drift because, in a sense, it's maintaining a memory and integrating changes on that memory that are going to start to drift, as you would expect any sort of integrator, particularly biological integrator. So the thing to notice here is you have a population of cells, and this is classic in biology, where you have cells that are tuned for a particular parameter. You don't find a neuron that indicates the angle all by itself. It uses a population individually tuned. And this has a lot of power and robustness with respect to damage. And that type of place coding, or population coding, is a hallmark of what you find in biology. And there's a lot of good reasons for that. So I did want to point out here, in most of this work, these are called head direction cells. But in most cases, it's really better thought in terms of body orientation, the direction of travel. Okay, the head can be looking all over the place, but for the most part, what's relevant here is the direction of travel, how that will affect it. So grid cells, these are the amazing cells. So this is the experiment. You have a square environment, a large, reasonably large square environment, where an animal will run around. And food is dropped in all kinds of locations in order to motivate the animal to move around. And so the gray traces are the trajectories of the rat moving around. And whenever a neuron, a particular neuron in the medium terminal cortex will fire, they will draw a dot. And this is what you get. As you find cells in the medium terminal cortex that fire repeatedly in this really beautiful hexagonal grid. And it's oriented obviously with respect to the environment. As you change where you're recording from, which neuron in a certain axis, because it's kind of a longish structure, you find cells that have different spatial frequencies. So you should be thinking, hey, wait a minute, this is starting to look like a 2D Fourier component, maybe. And so these are over here, these are just density plots, because if you have a lot of dots in one spot, they just line up and they disappear. And so the interesting thing, of course, is this hexagonal grid and the factor of this. And then place cells. This is actually, I'm presenting last, this is one of the very first things that was measured. So over here is the example for rat, two different big examples of place cells found in the rat. And notice that with respect to the environment, this is a pretty big, what we call a place field. And it's got reasonably long Gaussian-like tails. Right? Presumably the rat, one of the interesting things is that the rat actually this has a specific size. And what we now know is that the size can actually modify itself with respect to the specific environment. And over here is work done in Cindy's lab with postdoc Rachel Kulinovsky a number of years ago, where the very first measurements of place cells in the big brown bat was done. And I'll be showing you more of his work. And the exciting thing, of course, is that just like, this is a mammal, just like other mammals that have been studied, we're finding place cells. And so we're going to get back to this particular study. There's actually two exciting parts of this study. One, that they were found in the big brown bat also. And the catch here in this experiment is that this was done on a crawling bat, not a flying bat. So this is a crawling space, it's a place in which you can move around. In a semi 2 plus B sort of.
Timur, do you think people have all these kinds of cells too?
They do.
We have the same kinds of cells.
Yes, I don't remember exactly which experiment, how they were measured.
We've done recordings from
Epileptic patients.
virtual reality.
Yes. So that raises another question, are they exactly like this? And it turns out they're going to be similar, but not exactly. So the one thing I didn't tell you yet, that I find probably the most exciting thing here, the location of the brain where the place cells are found is known as the hippocampus. Actually, place cells are found in a lot of different places. But primarily, this is the big concentration, it's known as the hippocampus. And there's a number of, and it's a circuit, a loop circuit that has a number of different neurons in it. This same area in humans, based on fMRI studies, and other lesion experiments and deficits, are that this area is known to be very strongly associated with what we call episodic memory. And when humans have been studied, episodic memory has been about remembering the sequence of events or a particular thing, like what you eat for breakfast and where did you eat it, and do you remember this particular event? Very specific sorts of memories. They're very, very broad. They capture lots of different pieces that get stuck together, kind of like a binding memory. And so one of the questions is spatial navigation in rats somehow associated with episodic memory? Is this an episodic memory system, or is it a spatial navigation system? And so there must be some link between the properties of the two problems. Okay. And that's the teaser, really. So here's the other thing. I showed you kind of these statistical descriptions of what these neurons do and where they fire. The interesting thing is that if you actually have a local field potential, a low frequency measurement in these particular areas, you find oscillations. In the rat, in particular, in the hippocampus, you find that there's about approximately an 8 hertz sinusoidal measurement that you can take. And what you find is that neurons in the rat hippocampus are going to be firing actually at those frequencies, at 8 hertz. This is known as the theta band frequency. And theta is actually associated with a lot of different frequency bands. But for the rat and the hippocampus, theta is meant to represent approximately a couple of hertz surrounding 8 hertz or so. And so what's interesting is that the place cell, so if you have a place cell and it's got a place field, as the rat enters that place field, the spikes that come out of the place cell are going to fire at a very particular phase. And as it progresses through the field, you'll see it actually process or change what particular phase it will be found in. And this is this is 2 copies of the same thing, of the same data. This is the theta phase, so 0 to 360, and then repeating again so you can see the transitions between 0 and 360. What you should interpret is that there's this broad sort of diagonal here, where depending as the animal enters the field, it fires at a particular phase, and as it moves into the center of the field, the phase has shifted. So this is when the spike fires with respect to the surrounding oscillation. And so one of the things here is that, so this has driven a tremendous number of models that are oscillation-based about how do you get cells that fire at very specific frequencies and very specific phases, and what does it mean? On the whole, there are lots of ideas. Source of oscillations have been associated with some of the inputs and some of the natural frequencies that are found in hippocampus. But we'll get into some of that a little bit later. What it means exactly is another big question. One of the things that people associate theta with is the learning process. Yes?
So for the head cell and the place cell, for example, if an animal lives in one environment, and if you bring it to another very strange environment for the first time, does it fire when the head turns, like the head cell?
Yeah, so let me address the place cell answer to that question. On the whole, if you were to take a given neuron, a given place cell, it'll fire in a different environment as well. And it will also look like a place field, generally. About as fast as you can measure it, there's already a place field in this new environment. It'll fire in that environment, it'll fire in this environment. And for the most part, unless they really look identical, there will be no apparent relationship between the place field in one environment and the other environment. And so, in fact, it's known that it can contribute. It can remember being, if if you move them between two environments, it'll go back to being the place cell for this environment, and it'll be place field of so-and-so in this new environment. And they're as fast as you're able to measure. So it's an inherent property. You're not learning place fields, generally speaking. You are using them in some fashion.
And what about the rotation, the head rotation stuff? Does it also fire?
Yes, it will fire, and it will fire with respect to its other head direction, other cells as well. So if one is 20 degrees off of the other one in general, in the new environment, that relationship will stay still, but the orientation is arbitrary. You can move so this is a huge literature, so I'm kind of hesitating to get stuck in there too much. But the compass orientation with respect to the environment is defined by the sensory scene. So if you present a visual, if there's like one object visible there that's unique enough, then that'll be a reference. So if you move that visual target without any conflicting information, the rat's whole world rotates with that landmark. So it's clearly using its senses in order to figure out what the origin or the orientation is of the system. And then we'll move with the environment.
So you said that when you drop food on the place, then it goes and picks up and the head orientation and finds space. If you put it on a platform, they have the same or they will different. The rat doesn't move anywhere, but the platform only moves in the direction. So only the eye segments that for place cells because movement is not in place at all.
I'm not quite sure I understood that. So what's the platform? Is it very restricted?
Say a circular platform where it is rotating, the platform is rotating.
Well, the platform's restricted.
Okay. Instead of placing food so that the rat goes and picks up.
Oh, I see.
There is because I get the feedback from the cells from the body, then maybe the place
Right.
gets
Basically, there's mixed answers to that question, depending on the specific experiment that people have done. On the whole, one of the things that people have found is that the animal is not the one doing the navigation. You don't see much modulation in the way that we're talking about. Place cells can kind of go blah. They will fire not in a very precise way, unless the animal is actually actively exploring, moving itself. There's a lot of, and that's one of the experiments that really has shown that if you want to understand a lot of animal behavior and animal neurobiology, you have to really consider if the animal is doing it itself, if you're playing something like that, because it can be very different in response. And this has been seen in many different kinds of experiments. Yeah. Okay, so in rats, oscillations will be seen everywhere. It's a huge factor of it. You see very large amplitude there. But in bats, one of the notable issues that came up in the big brown bat hippocampal work was that there wasn't continuous data, raising all kinds of technical questions about, was this about an experiment, et cetera, et cetera. And after lots and lots of controls, lots of looking at it, essentially it's still held up. What you do see some data, but it comes and goes. It's transient for maybe a second or two. And so apparently, and yet you have place cells. So clearly place cells are not dependent on the presence of this oscillation, or at least there's a lot of, it raises the question whether or not you need theta in order to produce the place cell behavior. So this discovery basically triggered a whole series of new experiments, and that's what Malcolm is doing in his lab now in the Weizmann Institute. And so here, the other thing you notice is it's not exactly 8, but it's in that theta band range. And so there's nothing magical, particularly about 8 hertz. And it's very clearly not present, even though you see hash here. So Malcolm has gone off to his own lab, started his own lab, and has switched to studying primarily Rosettus egypticus, the Egyptian fruit bat. This is an echolocator, although of a different type. And this animal is known for some fairly long-range navigation in terms of its origin. And so, also in 2D place cells were found in the crawling Rosettus bat, also lacking continuous theta. And so, the big question, is there something about bats? Or is there something, quote, special about rats, in some sense? And we're mixing up two different things related to behavior and the construction of the cell properties. And so this is a raging, angry, sort of rancorous sort of debate right now. In fact, they came to our campus this summer and delivered some even more exciting new data. And had they been able to publish more of the data sooner, I would have had a more exciting story for you to share. So 2D place cells and then 2D grid cells have also been measured. Also without the theta, which normally you see theta in rat grid cells. And so what's interesting is that when we say theta only transiently, really it will turn out that it's associated temporally with vocalization, with sonar, with exploration. And in fact, in humans, this is something you also find, is that although there may be some theta deep in the background, on the onset of learning, or on the onset of trying to recall a memory, you see dynamic modulation in theta power in different regions. And so theta is somehow associated with the active process of learning, or possibly recalling. And the exciting thing, of course, is that what this implies, though, in the rat, is that as the rat's running around, it's maybe actively always trying to remember where it's been. And so it's in a slightly different mode. With bats, unless you vocalize, you're not going to get data about the environment. So we have kind of a mystery, why isn't theta continuous for bats? And then a larger question, which is the one about 3D. What is 3D? What are you going to see in place cells when we start to talk about flying? Bats are very, they operate in the 3D quite a lot. And the reason we have ONR funding is that it's also believed that this is relevant for aquatic creatures. So if you're a flying animal and you're subject to wind and such, and you're in a three-dimensional environment, you really have to worry about drifting away and estimating position. And also, this will also be true for a lot of marine mammals in particular, and fish and so forth. So this has been studied a lot in rat, well, not a lot, it's been studied in rats. And this is one of the notable pieces of work in the recent literature, which is, so people have studied rats not just in two-dimensional environments, but on ramps and quasi-two-dimensional environments, and those have come to a lot of criticism. This is one of the nicer ones recently, where a pegboard was created, where the rat had to crawl up and down, move around through the grid. And so the 3D, or the elevation component, was relevant to this particular behavior. And so as it moves up and down, you can record place cells, and you'll see grid cells in the next topic. So this is a lab work by Kate Jeffrey over in the UK. And in this example, what they find is they find place cells, but they're elongated. It's as if it considers even the vertical extent more or less as the same place. So it's not coding very tightly for vertical dimension. This one is not so bad, but the majority of the cells see some sort of elongation. You might think that the relevant resolution in this particular environment would be in the layers of these pegs. Okay, so even in this case, it spans multiples. When you go and you look at the grid cells, grid field, so in this example, it's not really traversing a two-dimensional arena in the horizontal plane. It's really more in a one-dimensional, but it's going vertical too. As you see, even more dramatic neglect of the vertical, essentially. And so this had suggested that maybe vertical is different, conceptually different for animals than the horizontal. And there's a lot of reasons to think that might be true. In the horizontal plane, if I'm a cat or a cheetah and I'm going to travel over to hunt somewhere, then in a sense, I can go a lot further. It goes on in infinitum, basically, in any direction that I choose to be. But vertically, it's different. There's the ground and there's higher, essentially. And I don't need to be able to code as I keep going up and up and up and up. Even bats that travel very high may not need that sort of system. So it may not just be another grid cell in three-dimensional plane. And that's the perspective I'm sort of taking. But what happens in that? So Malinowski has been doing these experiments and recently got data for Rosetta. The Egyptians proved that. And so he does find, the lab does find these somewhat 3D place cells. So this is an example where, sorry, I didn't explain the previous script. This is an arena where they created a pretend tree where they put fruit and other food items that the bat has to find. So we'll circle around the tree and try to find goodies. And when you do this, essentially, you can discover that there's a three-dimensional volume in a particular location in which this cell will fire. And more importantly, if you look through the population of recorded cells, you do find a variety of different locations, different sizes. So at least in the past, there's a suggestion that we are going to find 3D place cells. One question is, would we find three-dimensional grid cells? And where would that look like? And there's no answer yet. Soon they say. So let's focus on models. So I'm going to tell you about a model for head direction, and I'm going to talk about the grid cell model and give you a teaser for the place cells. And that's a relatively easy concept, conceptually. So there are two main camps with respect to grid cell models. I think the one on the right is easier to describe, so I'll do first. We'll do that one first. This is a 2D attractor. Now, the idea here is that I'm going to have a two-dimensional plane of neurons, physically, two-dimensional plane of neurons. And they're going to have a connectivity pattern that's going to be locally excitatory. They're going to support its neighbor's properties. But a little bit further away, I'm going to inhibit in a ring. And this is very classic. You find this sort of connectivity everywhere you look throughout the brain. And so this is not a stretch. When you do this and you excite everybody, but they all try to become active and they all try to suppress each other, what you get are bumps. So if it's a 1D structure, you'll see stripes. Okay? In this kind of a structure, you get these bumps. And what's the tightest way of packing bumps? Hexagonal close pack. Right? When you do oranges, basically. In a plane. And so this is an interesting model in the sense you have this two-dimensional plane of neurons, and they have specific connectivity that basically creates a ball-like structure. And when you activate the whole sheet, they're going to self-form into this hexagonal grid of bump activity. So I'm going to have this hexagonal grid of activity on the neural sheet itself. Okay? And so the interesting thing about this is that if you design it right, you have local excitatory feedback that's recurrent, you can maintain activity. It stays there. Okay. There's all kinds of issues that relate to this. There are a lot of neurons that are needed. Creating movement of the bump pattern is going to be tricky and requires specific wiring. If you look at a lot of the models, there's a lot of that crazy specific interconnectivity that is questionable whether that's going to be realistic to propose. It doesn't require any sort of oscillation, and that's the opposing model. You can insert oscillations if you want, and that's a competing model. The other model here is the oscillatory phase model, in which you have oscillations. You have consider two oscillators that are both running at 8 hertz. If they're in phase, the summation of these two, the inputs from these two oscillators, are going to come in phasically. And so a neuron may fire at 8 hertz. If one is running at 9 hertz, then they're going to phase lock every 1 hertz, 1 second. And so this group, what you can do now is by storing the relative phase, by shifting the phase of one, I can completely shift out of phase. It's 108 degrees out of phase, and it won't actually respond at all. A downstream neuron won't respond at all. If I shift the phase, as I keep going further, it's going to repeat again. And so this model uses the relative phases between oscillators to store information about position, but they're oscillating. And by arranging how these phases shift with respect to velocity and movement, you can make a pattern in the projection of this grid pattern. And what's interesting about this model is you don't have to build a two-dimensional array of neurons, of grid neurons, to have one grid neuron. You can build one if you wanted to. And so it's very efficient in terms of hardware, and that's a very attractive thing. One of the things you find in the grid cell patterns is that neighboring cells aren't exactly tightly coupled in phase. And that would suggest that this might actually be nice. This structure here, though, although you can create the hexagonal bumps using a very specific phase relationship between them, it's not automatic. You don't have to get that. So the question is, which one is right? What are the disadvantages of each? The other interesting thing is that the memory is stored in the phases here, not in the grid pattern here. In this model, the memory of location is stored in the grid. And if I have to build lots of different spatial frequencies, I'm going to need a lot of hardware here. I'm not going to need quite as much over there. But it has other things. So how do you go from grid to place? This is the classic sort of Fourier decomposition of a bump or a pulse. Is that with the right phases and a spread of frequencies, I can define a delta function somewhere. And so in this example given by Moser, if you just take these patterns, different spatial frequencies, they have a particular phase, add them up, you get this pattern. And if this is the input current to a neuron, and you have a threshold set to dark red, then you get a single location. So you can use the grid cells to create a single location response. Notice that this is much tighter than that is in general. I mean, it's not scaled the same. But it raises a number of interesting questions. Okay, so since I'm way lasting over time, I'm going to see if I can speed up a bit. So my lab does neuromorphic VLSI, and so one of the things we're trying to do is to model some of these networks. In general, neuromorphic VLSI really is about trying to understand the neurobiology and the neurocomputation that's going on, and building an electronic circuit, usually analog, that tries to mimic that basic computation. If you represent data differently from biology, if you represent data differently than biology, you're going to end up building a different computer than what biology is building. And it will have it may have the same results in ideal conditions, but will be very different as the signal quality degrades. So I'm going to skip through that. My student, Tarek Masood, worked on head direction cell system and the grid cell system. In general, the idea here is that you have neurons that respond to certain directions. And so the going model really is that although you find neurons that are not tightly coupled to each other physically, you don't see them, the next door neighbor neuron being one degree apart, you can arrange a model that's an attractor model, which is in a ring structure, that locally excites itself and its neighbors, but suppresses longer range neighbors. So the idea is that I can have a representation where a ring network has a group that are active, and that represents the current head direction. This is a memory and an integrator if we design the movement just right. So if you do the center surround type of connectivity, just like we said in the other case, the grids, in a ring structure you get stripes, and if you do a competitive network where only one bump is going to be active, you can enforce one group of neurons to represent an angle. And if you design a network that can allow it to turn, then you can move this around as the animal turns using the vestibular system. So we designed circuits using CMOS to mimic neurons. There's a membrane potential that sits on top of a membrane capacitance. There's different methods for getting current into the cell to excite the neuron. Once the voltage rises to a threshold, it will fire a digital pulse, effectively. And there are a lot of other properties like refractory periods and rate adaptation that we model sometimes. But this is a representation of the complexity of a given neuron circuit. And so we have synapse circuits that try to convert narrow impulse response action potentials into longer, more sophisticated current profiles over time. Obviously not going to go through that detail. And we design a chip, send it out. We use the Moses service in order to do that to the department. So Tarek designed a system using the chip and a PIC microcontroller to take the spikes and create the network architecture on the chip. So these are neuron spikes. You can't see the dots because they're so tightly packed together. This is a kind of a blow-up, where you have, these are different neurons in the ring, and then it wraps around to the top over here. You get these stripes. When you turn on the competition, then you eliminate the stripes, and you get that single bump activity. I'm going to skip how we do the movement. And the system we put together. But, so this is a movie created from recorded spiking data from the chip, where we are manually changing the velocities, making it turn around. And so then we took the addresses and created a little animation graphic for this. But the idea is that we have control over the speed of this bump around the ring. And that's a consequence of the particular way in which we implemented movement. So we can go both directions. And this is essentially taking velocity, angular velocity, and integrating it into position. And that's the idea. So if we stop getting an input, it basically stops and holds this estimate. So we have a wide dynamic range of control in terms of being able to make it spin around. For the most part, an animal generally won't be doing a lot of dynamic range spinning. It will turn at a particular rate, slower or faster, but not over a tremendous range. Inevitably, in any sort of integration system like this, there's going to be drift or error or leak. And so we were able to quantify some of this. And of course, this means that your estimate of position, your estimate of angle is going to drift over time. And it's known that it does in biology. But the saving grace is in fact that if you can remember that a certain direction corresponds to a certain sensed object in the world, you can phase reset. And this is a system that we did, we demonstrated. And it's believed that this is a very common process, not just for the head direction cell system, but for other systems. And we'll talk about with respect to mapping and position. So there's a system we designed. We used a sonar system that would rotate, and every time it would try to maintain an estimate of its position, and converting the potentiometer here into a velocity signal that we converted into spikes. And then we use sensed objects using sonar to reset the phase. And so it looks kind of like this. There's the blue is the true angle, and the black is the estimated angle. Sorry, it's the other way. Black is the true angle, blue is the estimated angle. It's starting to lag. And if you don't do any sort of correction based on sensed objects, it just gets worse and worse. And the particular chip has some mismatch that causes some of the movement signals to drop out, and thus it's lagging in this particular case. So if you use correction, I'm no surprise, you can use phase resetting essentially to, every time you encounter that object, you can fix the phase. So this is a little more detail. Just looking at the phase change over time. So as it goes around the circle, you see it drifting, and here we do resetting. So we attempted to do a neuromorphic grid cell system. One of the big problems with going to 2D now is that the one-dimensional connectivity pattern was pretty brutal in terms of how many spikes and what the connectivity we had to do. Two-dimensional is going to be n squared worse. So one of the things was going to be n squared instead of n connectivity. And so we decided to take on a different tack, which is to use more of an analog continuum for this kind of a circuit. So this is a repeat of the idea of the Fourier decomposition, that if you have enough frequencies, spatial frequencies, you can put them together to create a system. So we are using the two-dimensional grid, two-dimensional attractor model. And so this is a simulation where with a lot of noise added. By creating these cell structures, you get different spatial frequencies on your grid. We designed a two-dimensional array of 32 by 32 pixels using a current mode model for doing the center surround behavior. Let's get the details here. And so this is like the lowest spatial frequency, which is just put one big blob in the middle of the chip. And those are the spikes for the different neurons in the. And this actually is thought, the little holes in this blob is really thought to be more of a readout problem on the chip as opposed to a failure of the actual blob. And so we were playing with different spatial frequencies, and he was able to demonstrate different spatial frequencies. This has toroidal boundary conditions. So the neuron here is actually connected to the one up here in the current spread. So one of the directions we've been moving in, and I'll finish in about 5 minutes, is to try and accommodate a lot of the new map data. Can we build grid cells not using the two-dimensional attractor network, but to use more like the phase interference model, which has the benefit of lower hardware costs, which is attractive for us because we want to build many spatial frequencies. If I have to build a two-dimensional chip with tremendous amount of wiring, I'm going to have to repeat that many times. Here, because a single grid cell can be created using just 3 oscillators, and I can just add as many grid cells as I want to. Maybe I don't need to build a large array. It will save me a lot of effort. So what follows now is a MATLAB simulation of a new model that's based on the phase interference model in one aspect. The phase interference model works based on 2 things. One is the phase interference gives you a measure of the position, the translational speed, how it changes. And that's where the memory sits. And then secondarily, there's this mapping in these 3 cardinal planes that creates the grid structure itself. And so if you just keep the mapping, let's say the rat is moving or a bat is flying in this vector, you look at the projection of that vector onto these cardinal directions, and you rotate. So now, instead of just an oscillator, I'm going to create a ring oscillator, just like the head direction cell system. Now I'm going to rotate this thing around at a rate that's proportional to the translational speed along a particular direction. So I'm going to have a ring oscillator for 0 degrees, I'm going to have a ring oscillator for 120, and another one for 240. And they're going to, the bumps are going to spin around according to their value of projection onto these cardinal directions. Okay? And then I'm going to sum them up, or I'm going to do a nonlinear integration of these 3 angles onto a single cell. And in biology, it's not at all clear whether you're going to sum the contributions of the 3 oscillators or do more of a nonlinear multiplication. Because in a sense, what you're really after is something like a coincidence detection. And so you can do multiplication, you can fake multiplication by doing the sum of logs, as an example. Or you can have 3 dendrites that saturate with the inputs, so that any one dendritic tree cannot drive beyond a certain level. And if you design it so the threshold is like 2.5 inputs, two hard inputs won't fire the cell, but only 3 will. And so that's kind of what we've done here. And we've put it into VLSI as well. So this is just a pure simulation. So what you're going to see here, so this is nice, this is kind of framed in the middle here. So I have 4 spatial frequencies. Each spatial frequency requires 3 oscillators. So here are the 4 spatial frequencies, and I'll explain that in a second. So there are 12 oscillators here, and they're ring oscillators. And here I have kind of a Gaussian bump that rotates around. So when we see it run, you'll see these bands move back and forth. And the simulated mouse is going to go in a little circle. And as it goes in a circle, the rates on all of these rotational ring neurons are going to start, ring oscillators are going to start to change. These 3 oscillators basically project to grid cells. I've simulated a 15 by 15 array of grid cells. You don't really need to, you just need a handful. You probably only need about 10 to 12 grid cells in order to be able to do the next stage, which are play cells. But it's pleasant to be able to see the grid itself, so I've done this more visually for confirmation purposes, so it looks right. And so the main thing about the different spatial frequencies, and when we talk about space, we're not talking about in this space, we're talking about in the environment in space. Okay? So the spatial frequency, the way I get different spatial frequencies is I'm going to change the coupling between translation and phase rate, how fast they spin. So the top oscillators spin fast, and then slower and slower, and you'll see basically, so this is the neuron space, it will rotate more slowly, it will translate more slowly. So that's the model. Until it's now be a bat model. Just as an example. Okay, so I started at 0, 0 at this location, and basically went around and integrated all these phases, and yay, fortunately, they all come back to 0 again from the same location. So one of the things that we notice here is that all the information about position actually exists here. That's where the memory is. This is just a reflection of that memory. And notice, I'll run it again. At this point here, I plot a circle. And this will be relevant in the next section. It's at this time that I'm going to take a snapshot of all the grid cell patterns, which is to say, I'm going to record the phase of this grid cell pattern at that moment. So we have a circuit, a neuron chip that worked with TARC on. And so we have 3 synapses that basically saturate. These are post-extension synapses for those of you circuit people. And so this calculates essentially the coincidence of 3 inputs. And then we go to a conductance-based neuron and generate spines. So using the MATLAB simulation of the ring oscillators, the computer then spits out address events to the neuron chip, and we are able to reconstruct grid cell patterns on the chip for a single grid cell. That's the interesting thing, is I don't have to simulate the whole 2-dimensional array of neurons in order to get a single grid cell. And so this is kind of a beneficial aspect of this structure. And so when you take that snapshot of cells, and then you actually move the mouse around, and you see do I get a place cell? Essentially, you're cross-correlating the grid pattern that you stored and the grid pattern that you see. And when you do that, and you sum them all, essentially you get a giant peak right where you measured it. You threshold it, and you get your place cell. So this can be a very efficient way of constructing a place cell. So this is really kind of the last piece, is that how do we get to 3D? So I told you my unsubstantiated claim about height being a little bit different. But there's a lot of reasons to think that that might be true. But the way to do that here is really you need a place cell population representation of height, and you do a correlation between, a coincidence between the grid cell structures and the height variable. A 4-way coincidence, and essentially you get 3-dimensional place cell. So we're developing that and coupling with our sonar systems to provide the velocity signals. And hope to demonstrate some sonar-based hovering control, position control in the relatively near future. So with that, I will stop.
Excellent. You have time for a few questions.
So Timur, maybe this is diving a little too deep, but you mentioned in one of the recent circuits that you put up that you were using a conducting space neuron. Can you say a few words about why you use a voltage versus conducting space neuron?
It doesn't matter. You can do it either way. Conductive space neurons are considered to be significantly more realistic from a neuron perspective. With conductives, you can do a lot of other things that you can't do with voltage-based sort of neurons. Conductive space basically, instead of just injecting current, what we're doing is we're changing the conductance between the membrane potential and some other reference voltage. This actually allows you to have both excitation and inhibition, which I want to show, simultaneously in order to compute a ratio. And this ratio is something, it's a division that you can get. You can do division with synapses instead of just doing addition subtraction. You have to resort to your log transformations. And the other thing about conductance is that the capacitance in the neuron no longer acts as an integrator per se. The answer of whether the neuron's going to fire or not fire is mostly determined by the conductance values, and the capacitor just slows things down. As you end up like a resistor divider, the capacitor just sets a time constant, which could be relevant in computation, how long it takes to get there. But it doesn't, so one of the problems with a lot of voltage-based neurons is that if you have some residual voltage from a previous calculation, that now affects the integration as to whether you're going to fire or not fire, because it's a bias in the integration. And with the conductance space system, you get rid of that. It just affects your latency, but it doesn't affect the answer, which is really important.
John.
Yeah, I was just going to ask two questions here. One is, why don't you go for a 3D interactive model? 2D interactive, why are you studying
These
oscillations?
are 3D, it's insane right now.
All right, okay. So,
But, no, it's something to consider.
I mean, I'm just curious about how you could actualize it. I mean, I thought it was a very clever and creative way of approaching it, oscillation mode, but
So one of the reasons I got interested in the oscillator models, again, after rejecting them initially, is the fact that the 2D interactive model that we built with time, the mismatch in the transistors and the structures ended up, if you think of this as an energy landscape, is that it's very, very bumpy because of the mismatch. The different neurons have different coupling coefficients, even though they're not supposed to. And you put an activity pattern on there that's supposed to be, that's related to each other, and you try to move it across this landscape, essentially the bump pattern gets stuck. And in fact, although we designed it with the ability to move the 2D grid pattern on our chip, it was very hard to do. In fact, as far as anybody else is concerned, we got it to move, but as far as anybody else is concerned, it's not easy to move at all, in spite of all the complex things we had put into it. When you look at how to eliminate all that mismatch, I'm left thinking about the biology, is that unless there's a really good way of learning out, well, first of all, the movement network is going to be insane for even the 2D case. It's pretty crazy. And if you have to compensate for mismatches between neurons, which frankly is going to have to be done, it makes it a very hard problem. And one of the cool things about this system, it doesn't have that problem. I can shift an oscillator phase and it will move. It may not go in the right place, but it will definitely move. It will change its characteristics. And so there's nothing to say that there isn't a model that's halfway between those two. You can build a grid cell pattern using the oscillator phase interference model, and then they can be coupled to get the benefits of the attractive model without necessarily having the cost to build it.
yeah, so the second part was, the data that you showed from the English group where the mouse didn't really care about the vertical dimension. May reflect the fact that there's not that much interesting happening in vertical dimensions, but if you live in a rainforest with a canopy where there's very different kinds of fruit and different levels,
So there's something to remember.
something to remember, then
Right.
it all of a sudden becomes useful.
Absolutely, and we know that the experiment dramatically changes the results in many, many cases. And this is one of the cases where if you change the scale of the vertical dimension, you can see some of these things scaling with it, which is kind of what you'd like, but it's fun to consider how that can be done. In the phase interference model, it's easy. You just change the translation between translation and phase rate, and then you put scaling on that, and it scales across all the space. Whereas in 2D model, you actually have to change the activity. So it's a little bit.
So were each of the three oscillators associated with one of the cardinal directions? Is that what you said?
Yes.
Okay. My other question was, earlier you were talking about how there could be a link because they're both associated with the hippocampus between spatial navigation and episodic memory. And I know from my previous readings, I guess, that high levels of stress can disrupt episodic memory based on kind of shrinkage of the hippocampus. Do you think that if it was proven that spatial navigation ability was kind of decreased by high stress, that could further kind of link the two in animals?
The problem with stress as in an experiment, of course, is that it never affects only one thing. And so it makes it very experimentally difficult to actually prove anything. Having said that, I don't actually know if that's been true. They don't study episodic memory in rats generally, and they don't study spatial navigation so much in humans, although it's definitely something that's happening now. I mean, they study behaviorally, but they don't study being able to record from neurons or even actively in fMRI. Although there's a lot of virtual kind of mental map kind of experiments that people do. But I don't actually know the answer to that.
So in the beginning, you mentioned the motivation from robotics. Is there any implication now about like navigation, maybe in GPS denied environment?
Absolutely. I mean, in the sense that you don't have to build it the same way. The biology has to do it. I'm not sure that there's any lessons specifically here that you wouldn't have thought necessarily in engineering terms. It's not clear at all what the benefit of having hexagonal grid is specifically versus a Cartesian grid. I'm not actually sure that that's a particular benefit. It has some benefits in terms of tessellation of spaces. You can actually get finer tessellation of space. And things are more round than they are square in natural environments, but not so in human environments. I think maybe if there's anything, it probably more relates to the representation of space. So one of the questions that has come up with this grid, head direction cells feeding grid feeding place, is that all the information is actually already there in the oscillator model. And why would you need to create grids to create place? Well, presumably, it's technically possible just to go from the oscillator straight to place. And the reason people have argued for these two separate representations is that it's easier to build the next stage, which is to associate a memory with a place cell, because it's a group of cells that are just firing, and no other neurons are firing. And so it's easy to do a linking between attributes of a place or the link between places, which is about navigation. If you have just a single group of cells firing, this place cell representation. On the other hand, the grid cells are very much about mapping, calculating metrical distances, because they're very good at, because of the repetition, it maintains kind of this fixed metric step that you're making as you travel. And so that's the argument that people have made, why you might have both for solving problems.
Let's follow up on that. What would be, for this navigation system, what do you think, like in a 2D system, how many number of neurons would be able to mimic the system for a real place?
So then it becomes all a question of resolution. Right? If you want finer resolution, you need more spatial scales, which is a classic Fourier trade-off. So no, I don't have a specific answer for you. Compared to what, right? Two 16-bit registers with X and Y. You know, so it's not a really fair comparison to make at this point in that sense. One of the things about neurobiology and the whole approach of neurobiology really is that a tremendous amount of redundancy. And I talked about the population response. If a neuron dies, you just have a slight deficit for that value. You haven't killed the whole thing. If your 16-bit register that stores X is dead, you dropped a dimension, right? And so it's a very different kind of system. And one of the things, of course, neurobiology has evolved in the context of is damage, brain cell loss. And so the whole, I think, one of the keys to survival, which includes things like, you know, NASA spacecraft, is that you can't design a system where failure of one module is going to kill the whole system. And so representations in biology seems to suggest that's true. If you have different neurons that represent different values, not only can you have one value, you can actually have two values. You can represent a probability distribution using that representation. You can calculate things like entropy, certainty of your knowledge information. So the representation that you pick can be tremendously powerful in terms of doing things that you didn't really expect. So we keep an open mind for that.
Okay, let's thank our speaker one more time.